Hello, everyone, and welcome to the Anti-Doping Podcast, brought to you by the Partnership for Clean Competition. I'm your host, Dr. Marie McNeely, and today we are joined by our guest, Dr. Brad Johnson. Listeners, Brad is the Gordon W. Davis Regent Chair in Meat Science and Muscle Biology and professor in the Department of Animal and Food Sciences in the College of Agricultural Sciences and Natural Resources at Texas Tech University. Today, we look forward to discussing his research on muscle growth and development in meat animals and potential contamination of meat relevant for anti-doping. So, Brad, welcome to the show today. How are you?
I'm doing great, Marie, and thanks for having me as a guest on your podcast.
Well, we are so excited to have you here with us, and we're looking forward to learning more about you and your research. And perhaps we can start with your story, Brad. Can you tell us, how did you initially get interested in science, and perhaps what path did you take to get to your current positions there at Texas Tech?
So it's interesting. I have a background in animal science from an education standpoint, but in fact, I've been around animal production my entire life. I grew up on a farm in South Dakota, and I was the youngest of 6 kids, and I was a first-generation college student. So I always knew I wanted to go into animal science and further my knowledge of animal production in that way. So I did my undergrad at South Dakota State University, and I was very intrigued by the real basic nature of how animals grow. At the same time, I'd always thought I would go down the veterinary medicine path, and I applied to vet school at the University of Minnesota, the adjacent state to South Dakota, and was accepted to vet school. But I deferred for some personal reasons and then started graduate school at the University of Minnesota in a real basic skeletal muscle biology lab and just really loved it. In fact, when I was an undergrad at South Dakota State, is where I had an opportunity to do a couple undergrad research projects. So I was always turned on by just the quest for more knowledge and understanding, especially basic mechanistic things and how that applied to animal agriculture. So once I got into graduate school at the University of Minnesota, I decided not to go to vet school, and I stayed and completed a master's and then a doctorate, a PhD in animal science. So I've always, again, been exposed to science on agriculture mostly, but it's really neat now to I call this transdisciplinary work, where I can apply some of my knowledge on the animal agricultural side to a different discipline like anti-doping.
I think that's really cool. And I think you get to be able to answer a lot of cool questions working in this transdisciplinary area that you're working in. So let's talk about your roles at Texas Tech University. We mentioned your positions in the introduction, but can you tell us a little bit more about what you do and perhaps share the broad scope of the kinds of research that you do in your laboratory there?
Sure. So there's 2 major focal points that I've worked on for several years as an academician. So prior to coming to Texas Tech, I've been at Texas Tech University almost 17 years in this chair position. But prior to that, I was assistant and associate professor at Kansas State University and basically did similar research at that location as well. So 2 major themes of research in our lab group, and the one that probably has the most relevance to anti-doping. As I said in my introduction, I'm trained as a basic skeletal muscle growth and development biologist. So really, it's more physiology and biology of how certain factors affect skeletal muscle growth, both during the embryonic phase, where we establish a number of muscle fibers, for example, but more importantly, from a meat animal production standpoint, being in an animal science department, a meat science group, we're interested in postnatal muscle growth and factors that impact that. So in fact, what's kind of led us to this crossroads of animal science and, say, anti-doping is that for many years in the United States, we've used what we call exogenous growth promotants to improve the efficiency of growth of meat animals, namely the efficiency of skeletal muscle growth or lean tissue deposition, the important protein component and all the important trace elements that are in that muscle. So since 1956, we've had the ability to use steroidal implants or anabolic steroids that are put into compressed implant pellets to enhance the efficiency of muscle growth in beef cattle. So coming up on almost 70 years of using that technology to produce safe and wholesome meat in the United States. So my whole theme for over 30 years of research has been using these exogenous growth promotants like anabolic steroids and also another class of compounds called beta-adrenergic agonists. I've used those to unravel the mechanism of how muscle grows, especially in postnatal muscle growth after birth. And really, my whole goal is to learn the fundamentals of this. So maybe we can do this naturally through genetic selection, other manipulation techniques, for example, to do that. So I've been working in this area for over 30 years of understanding how something like anabolic steroids can actually increase postnatal muscle growth. The other major area of research focus that probably isn't so related to anti-doping, very much related to meat science and meat quality, is when individuals say go to purchase beef or go to a high-end steakhouse for a special occasion, often the more expensive cuts of meat have more what we call intramuscular fat within the muscle, and that's called marbling. And these are lipid droplets that form outside of the muscle bundles and are very important from a meat quality standpoint. So our lab also is equally interested in knowing factors and the mechanisms of how we get more of this marbling in the muscle that increases meat quality. The United States is known for high-quality meat, especially high-quality beef. So much of our high-quality beef has a lot of this marbling. We end up exporting that to countries like Japan and South Korea and now China.
Very interesting. And I think in hindsight, when you paint the picture, it all makes sense, this connection between animal science and anti-doping science. But I think there are so many researchers doing great basic and applied research that could be relevant for anti-doping, but people don't necessarily make that connection. So for you, Brad, how did you first get introduced to this field of anti-doping and discover that your particular expertise was relevant for the field?
My very first faculty position was back at my alma mater of South Dakota State University, and it was very applied. It had an extension component where I helped local cattle producers in South Dakota. I had a heavy extension appointment. But because of my basic muscle biology background with my dissertation research at the University of Minnesota, specifically with anabolic steroids and the big one being trenbolone acetate, a synthetic androgen, I'll never forget this, and I think I have a hard copy of this email stuck away in a file cabinet somewhere, but a publishing company in New York City that was publishing back then a glossy human muscle bodybuilding magazine reached out to me to write a guest editorial article on the use of trenbolone, which would have been an illegal use of it, to enhance muscle mass in human specimens. And I obviously declined to do that because it was illegal, but that was probably in the 1997, 1998. And I'm thinking, why would anybody want to do this? So I've always had that in the back of my mind. But now let's fast forward to where I really got engaged in the anti-doping side. Would have been about 8 years ago exactly. It would have been the fall of 2016. And I remember I received a phone call, kind of a cold call, from a couple individuals at USADA. They had a case that involved trenbolone acetate, which is a very commonly used substance in beef cattle production in the U.S. as well as many other countries worldwide. And that was kind of my first exposure to working in the anti-doping area. What's interesting is, coincidentally, during the same time frame, so that was in the fall of 2016, early 2017, I was appointed to an expert committee that's called JECFA. That's a Joint Expert Committee of Food Additives. And this is a joint committee between the FAO, which is a Food and Agricultural Organization out of Rome, and the World Health Organization out of Geneva. And I'm on the Veterinary Residues Committee that establishes basically maximum residue levels for different veterinary compounds that are used for animal production worldwide, including some of these compounds like anabolic steroids that are used for growth promotants in beef cattle and other species, as well as other classes of compounds like the beta-adrenergic agonists. So what's interesting is those are 5-year appointments. I was reappointed a couple years ago to that veterinary residue subcommittee. But the knowledge base of knowing about the residue limits, the maximum residue limits set by an organization like JECFA for Codex, which Codex is responsible for global free trade standards. That's been very beneficial to bring those two groups' thought processes together of anti-doping as well as Codex fair trade standards, and use the knowledge that we have from establishing the maximum residue levels through codex and using that to apply to the anti-doping space as well. So both of those things kind of happened in 2016 and 2017. So that kind of put me on a different trajectory as well from a research and a consulting standpoint.
That's really interesting, Brad. And I love hearing this often serendipitous series of events that ultimately bring people into anti-doping research. And for our listeners out there who might not be very familiar with animal science or perhaps this intersection with animal science and anti-doping, can you share some of the key background points for why some of the practices surrounding livestock management and meat processing are particularly relevant for human anti-doping?
As I said earlier, Marie, we've had the ability in the United States to use anabolic steroid compounds in these special steroid, they're called implants or compressed pellets that we put in the back of cattle's ears. We've been doing that practice since 1956 in the U.S., so nearly 70 years. So really the importance of where animal science and meat production kind of intersects with anti-doping is that our U.S. Food and Drug Administration has a whole list of products that have been approved by their organization to be used to be growth promotants to produce safe, wholesome food in the United States. So since 2016, I've been involved in probably over almost 24 different cases. Well over a third, close to half of those cases have involved one particular compound called trenbolone acetate. And trenbolone acetate is a synthetic androgen, very similar to testosterone, but it's a very potent anabolic agent that was approved to be used in meat animal production in the U.S. in 1987. And this compound is also available on the black market. If you search the internet for trenbolone, you're going to find all kinds of vendors that are willing to provide this to be used illegally as a muscle-enhancing agent in humans as well. And just to put it in perspective for the listeners, at any given time in the U.S., we probably have close to 10 to 11 million head of cattle that are in feedlots that are being fed for meat production. And at any given time, 90 to 95% of those cattle probably during their lifetime receive at least one trenbolone acetate containing implant, and sometimes they can actually receive 2 or 3 of those during different production phases. So that is where the intersection occurs, is that we have this approved product named something like trenbolone acetate, and we can administer that as these implants in the back of the ear of cattle during their growth phase. And there's an opportunity, obviously, to detect residues of that compound at harvest in the meat that is produced from those animals. And now certain athletes have said that they have an atypical finding or have failed a urinalysis for trenbolone. Usually it's epitrenbolone, 17-alpha-trenbolone in their urine due to consuming meat that potentially had high enough residue levels of trenbolone 17-beta in the muscle or liver or other offal items like kidney and liver, that is the reason why they failed a particular test. So again, there's other compounds that are approved, namely a class of compounds called beta-adrenergic agonists. These have been approved not only for cattle in the U.S., but also for pork production or swine production. And these are compounds such as ractopamine, zilpaterol, and lobavagran. And so these are also compounds that have come up in certain anti-doping cases where individuals might fail a zilpaterol test and have blamed meat consumption for that. In addition to the U.S., many of these same compounds that I've talked about, Marie, are also approved and used in many other countries across the world, like Mexico and Canada and South Africa and Australia and other Central American countries as well. So the international aspect of this becomes very important as well.
Definitely. And I think that's a really important point because athletes are often traveling for international competitions, eating local food while they're there. So can you talk about how this international aspect of it has maybe shaped some of the ways that you think about this problem?
Yeah, and this is one area that I think athletes really need to at least have knowledge of where they're going. You bring up eating local foods. And for example, certain Latin American countries use other compounds that are not approved in the United States that are either legally used in these countries, or there's even some compounds that are illegally used And for example, boldenone is approved as a growth promotant for meat animal production in many Latin American countries. Boldenone is a synthetic androgen similar to trenbolone, is not approved in the United States. This is also an injectable form. It's a little different than the implant. So, for example, certain countries like Mexico, Colombia, Dominican Republic would allow the use of boldenone legally. The big problem, and really what brought the whole meat contamination issue into anti-doping, was the compound clenbuterol. Clenbuterol is a beta-2 adrenergic agonist that I made reference to earlier. Clenbuterol has never been approved as a legal growth promotant for meat animal production anywhere in the world. However, we still see black market illegal use of this substance in many countries for meat animal production. And many of your founding partners in PCC on the professional athlete side are always concerned when they go into foreign countries about one particular compound, that being clenbuterol. And in fact, even USADA and WADA now have put some threshold limits on clenbuterol because certain countries, even though it's never been approved, it's still illegally used for meat animal production. And that's a compound that I'm very concerned about, both from a general human health standpoint, but also for athletes that may unknowingly go to countries, eat local food that could be tainted with some of these illegal substances.
Well, Brad, I think this background is tremendously helpful. And I think it's interesting. You mentioned your involvement in some of these anti-doping cases. And I think not many scientists are called upon to help in the legal sphere. So what has it been like in general for you to share your expertise in these anti-doping cases?
It's been very exciting, to be honest with you. And I shared some personal stories recently at the USADA Science Symposium with some of the other attendees that I've been a faculty member for nearly 30 years, and I've been doing research in this field for 35 years. And sometimes you need to get re-energized, right? And there's different ways to do that. You can do a sabbatical. You can kind of change your focus of research. You can start teaching different classes. But what really re-energized me was getting involved in some of these anti-doping cases at a legal level and seeing how, hey, we need more research in this particular area to both help from a legal standpoint, but also help on the athlete side, because there may be issues with some of these compounds entering the food system. So that kind of pushed me into a different research area. But the legal side, being called as an expert witness in many of these cases, has been very exciting and has got me thinking kind of outside the box, so to speak, and has pushed me down a different path of research that has been very fruitful and very exciting.
Definitely. You've been doing some phenomenal research. And Brad, I'd like to talk about some of your anti-doping related projects specifically. I know you were awarded a research grant from the Partnership for Clean Competition to study potential residues of prohibited anabolic agents in the meat supply chain. So let's talk about this project in a little bit more detail. Can you first explain your rationale for why you wanted to do this particular project and perhaps what you thought you might find?
This kind of goes back to what we just talked about, is that as we would prepare for some of these anti-doping cases on the legal side, as I would prepare my expert testimony and documentation for that, we had a few sources of information. So as I stated earlier, the FDA approves these products to be used for meat production that is ultimately then a source of human food, but they rely on the U.S. Department of Agriculture to monitor to make sure we do not have any violative residues of these compounds in the U.S. food supply. Also, over top of that, from an international perspective, I've talked about Codex and JECFA applying maximum residue levels of these. So we kind of have some benchmarks. We kind of have some threshold levels that we need to make sure that the residue levels of these compounds are under that. But we really didn't have kind of a broad scale representation of what is the U.S. meat supply at a retail level. The average person walking into a grocery store, purchasing beef or pork or poultry, what can we expect for everyday residue levels of these approved compounds, at least in the United States? So we felt that was really two main objectives. This would be very beneficial from an anti-doping standpoint to kind of establish what is a realistic level or threshold to expect of some of these compounds in different meat sources like beef, pork, and poultry. But also, I think it was important from just a general human food safety standpoint to make sure that we're well below some of these thresholds. So that was one of the main objectives, is we wanted to kind of do a surveillance of the U.S. retail supply of different meats like beef, pork, and poultry. So that was the main objective of this grant, really more surveillance work and establishing what kind of realistic levels of these compounds were.
That makes sense. So can you perhaps walk us through what were the methods that you used for this study and how was it conducted?
Again, I had a master's student. His name was Cade Sneathan, and he's actually defended his thesis, is graduating this week from Texas Tech University with his master's and then going on to Purdue University for a PhD. But what we did is we wanted to cover basically all the geographies of the United States. So we went to 8 different cities across the United States, and we ended up sampling at 48 different retail stores, all the way from very large retail stores that we all know very well to very small, almost ethnic type retail outlets in different cities across the U.S. So we had 8 different cities. We sampled 48 different retail outlets, so averaged about 6 different retail outlets per city. So my graduate student purchased a random sampling of different cuts of beef, pork, and poultry at these outlets. He would have traveled to these places personally, and then we would have shipped those back on dry ice to our lab here at Texas Tech. We then processed those, basically homogenized the samples. Most of our samples were either muscle cuts, but we also purchased some liver samples as well as some kidney samples at different outlets as well, or different retail stores. Once we processed these samples, our funding from PCC was a collaboration of not only Texas Tech University, but Dr. Matt Fedorik at USADA was involved, as well as Brian Aherns at the UCLA Olympic Testing Lab in Los Angeles. So we would have shipped our samples to UCLA, and Brian's group then would have done all the analysis for not only some of these compounds we've already talked about. So we tested for trenbolone and ractopamine and estradiol, but we also were very interested in seeing if some of these other growth promotants that have come up on the human anti-doping side, like the SARMs, the selective androgenic receptor modulators, if we could detect any of those compounds in the U.S. meat supply as well. So in addition to testing for compounds that we know that are used legally in the U.S. for meat production, we also tested for several ones that are illegal, like the SARMs, and we also included in their testing for clenbuterol that I made reference to earlier.
Well, Brad, I think this is a really exciting and really important research study. And I know no research project is without challenges. Whenever I hear scientists talk about their work, it always sounds everything's very linear, A to B to C. This is the steps that we took to do it. But can you talk us through what were some of the challenges that you encountered and had to work through in this particular project?
Well, originally we wanted to do this at the meat processing level. And we knew we were going to encounter some challenges of getting in and having to sign a bunch of affidavits. And so we decided to go the retail route, which was probably more realistic for the average consumer of meat anyway. Some things that getting some potential positive control or internal standards for Brian's group at UCLA. We had to find some muscle cuts that we knew were going to be negative for all these compounds, even the approved ones. So there were some challenges of finding samples to use as internal controls and things like that, but we were able to overcome all that. So outside of just the logistics of traveling and making sure we had enough dry ice and being able to find dry ice in some of these areas to pack and ship our samples back to the lab, we were able to overcome almost all the challenges.
Excellent. So let's talk about results. I think getting to this point where you have the results is an achievement in and of itself. But what were the results and findings from this particular study, Brad?
I might just focus on a couple items. Of those 48 different retail stores, for example, we had 41 muscle cuts of beef that we selected across 8 different cities. And I've been talking about trenbolone and a common growth promoter for beef cattle production in the U.S. So of those 41 muscle cuts, we had 4 positive samples for Trenbolone 17 beta, and these were at very low levels, okay? So we were right above our limit of detection or limit of quantification for Trenbolone 17 beta. So of those 41 cuts, those 4 that we were able to detect 17 beta Trenbolone, the average was 0.06 nanograms per gram. So that would be at a part per billion. That'd be the same as microgram per kilogram. So to put that in perspective, roughly 9% of the samples were positive for Trenbolone. Those 4 average 0.06 nanograms per gram. The MRL set forth by Codex or JECFA as being the maximum residue limit for muscle of animals that have been exposed to Trenbolone is actually 2 nanograms per gram, or 2 parts per billion. So you can see that those 4 samples that were positive were well below the limit set forth by JECFA. So how this helps with anti-doping, again, when I go in as an expert to prepare documentation as an expert witness, I often use that 2 parts per billion. If an individual claims they ate a ribeye steak, for example, I'll use that 2 parts per billion as kind of an upper threshold. But now knowing that a very small percentage of the retail beef actually had detectable levels and the average was under 0.1 nanograms per gram, well below the 2 nanogram per gram threshold, this becomes really important, I think, in understanding that we don't have a widespread abuse of this compound in the U.S. that is potentially giving us residues above the 2 nanogram per gram. We also found some positives for ractopamine in the beef supply, but not in the pork supply. We had 1 pig kidney that tested positive for nandrolone. So nandrolone is another synthetic androgen similar to boldenone and Trenbolone that has been illegally used by athletes. But what's interesting is pigs are very unique in that male pigs that are not castrated, they're called boars, have the ability to synthesize 19-norandrosterone, which the metabolite of nandrolone is 19-norandrosterone. So there has been some anti-doping cases revolving around potential 19-norandrosterone contamination from intact male pork consumption. So we found 1 pig kidney that actually was positive for nandrolone, which would tell us it was probably an intact male that was producing 19-norandrosterone. So that was interesting. The poultry supply was very clean. There are no exogenous growth promotants approved for broilers or poultry production in the U.S., and we did not see any illegal substances being used really in any of the muscle cuts or liver or kidneys, with the exception of the 1 pork kidney that tested positive for nandrolone, but we could explain that through the 19-norandrosterone stories.
Very interesting. Were there any surprises or things that you saw that you couldn't really readily explain?
Really, the most surprising one would have been the pork kidney, which we can explain biologically if it was an intact male. Again, as you remember earlier on, I talked about one of the major objectives. We wanted to make sure that some of these SARMs or SERMs, the selective androgen or estrogen receptor modulators that have found their way on the anti-doping front from a human use standpoint, I was confident livestock producers in the U.S. were not using those, but I wanted to confirm that. We found no residues of SARMs or SERMs in the meat samples that we tested.
I think that's reassuring. I think a lot of these results are sort of maybe confirming your suspicions, but let's talk about the impacts then, Brad. How do you think this line of research or the results from this particular study could impact or maybe enhance anti-doping efforts?
I think once we get this research published, and we're just on the cusp of submitting this manuscript in the next week or so, but I think it'll give us another reference point to say that, at least in the U.S., where we know we're using something like Trenbolone at a fairly high level in beef production, that it's very safe and that the residues are well below those limits set by JECFA or the U.S. Food and Drug Administration from a tolerance level standpoint. So it will kind of give us a snapshot of a realistic expectation that maybe 10% of the samples we could detect a residue, but it's going to be well below in that zero point 0.1 nanogram per gram or zero point 0.1 part per billion range. So I think we can use those numbers then to kind of do some assumptions when these athletes do test positive for these compounds like trenbolone, and kind of use that to say, hey, they would have to consume X pounds of beef at this level of residue at 0.1, but then we can also use the 2 nanogram per gram residue as well. So it's just another tool in our toolbox to use from an anti-doping standpoint.
Absolutely. I think having this data is tremendously valuable. So let's talk about what's next. Brad, can you share what are the next steps for this particular line of research?
Yeah, so interestingly, this is retail. The majority of the meat contamination cases that I've been involved in from an anti-doping perspective, Marie, are really at the food service level. So most of that meat would be purchased through what we call purveyors or distributors, where they would buy it directly from the meat processing companies. But there have been some retail cases where individuals have went out and bought their own meat at retail levels and cooked that as well. So we need to be cognizant that some of these cases that we've been exposed to on the anti-doping side already have been more at the food service level, at the restaurant level, more so than the retail. The other thing is that's interesting in the U.S., some of our more recent cases, the meat has been purchased and consumed from, say, a U.S. restaurant, but the source of the meat has been international. As I said earlier, we produce the highest quality beef in the world in the United States, and other countries sometimes would rather pay us more money than our own U.S. consumers would to have us export that high quality beef to other countries. So in lieu of that, we're bringing in lower quality meat from other countries. We're importing that into the United States. One thing that we've talked about, our core group that were on the grant, Dr. Fedorik, Brian Aherns, and myself, we would like to further this research by trying to find either at the food service level or the retail level to try to find product that we know has been imported into the United States from another country and test that meat. This is difficult because sometimes, because of the protection of country, not all the products are labeled with their country of origin. But a lot of times on the food service side, those boxes of meat that come into these restaurants are labeled with country of origin. So I think we're going to try to augment this research by specifically selecting imported products, especially imported beef, and testing those samples as well. And that will kind of shed light on allowing us to get more information for individuals that may be traveling internationally as well and have more meaningful data for them as well.
Absolutely. And I think a point that you brought up earlier, Brad, is your research is relevant not just for anti-doping, but also general food safety. So do you have thoughts for listeners out there on what measures could be taken to help, whether it's athletes or just other consumers, better understand what is or is not in the food that they're eating, whether they're eating out at a restaurant or just buying things at their local grocery store?
So many times over the last 8 years, people have asked me now, I've even had individual athletes reach out to me. So if an individual athlete reaches out to me and says, I've always been clean, but I also like to eat meat because of the high protein and the high trace element contents, very bioavailable source of zinc and all the B vitamins and high protein. What can I do to assure that I'm not exposing myself unknowingly to levels of some of these compounds? I don't want to be exposed to any of these. So the great thing about the U.S. meat industry is not only are we produce some of the most high quality meat in the world, we also probably have the most diverse production systems in the United States as any other country. So there are great opportunities in the U.S. to purchase any meat, but let's focus on beef that has been produced under all natural rearing conditions where they cannot use any exogenous growth promotants like trenbolone that we've talked about or the beta-adrenergic agonists. And in fact, certain meat processing companies in the U.S. only harvest animals that are referred to as all natural. So I tell athletes that you can buy this in retail, but also many restaurants, many food services now will have brands of beef like certified Angus beef, which there's a natural line of certified Angus beef where none of the animals have never been exposed to steroidal implants or beta agonists. So that's just a level of assurance, not only for athletes, but for the average consumer, that if you want to limit your exposure to some of these approved compounds, I would recommend going a route of selecting meat that is from natural beef production. In fact, one of our anti-doping cases a few years ago, the individual claimed they ate a particular cut of meat. This was at a restaurant. It was sourced from a meat processing company that only processes all natural beef cattle. This was a trenbolone case as well. The likelihood of trenbolone being in that cut of meat was near zero because this particular company tests and signs affidavits continuously proving that they're an all-natural beef provider. So there are many choices available, at least in the United States, for meat that has been produced under natural situations where animals have never been exposed to these compounds.
That makes sense. And I think the sourcing is so important, but people might be wondering also, are there cooking practices or preparation practices that can help break down these compounds that may be in the meat? Or is this something where the source is really the only thing that you can control?
What ultimately causes these issues, Marie, is that the active ingredient of some of these compounds, and we'll use trenbolone again as an example, the active ingredient is 17-beta trenbolone and will actually bind to different types of steroid receptors in these target tissues like skeletal muscle, adipose tissue, liver, and kidney. So it's that binding to the receptor, either the intracellular classical androgen receptor or a membrane-bound G protein-coupled receptor. So once that happens, these steroids are very durable, and even cooking methods are probably, for the most part, not going to degrade some of these anabolic steroids or even the beta adrenergic agonists. But the other thing that's important for the listeners to understand is that there's a fundamental difference in how we absorb some of these compounds. So the other thing that we use on the anti-doping side is that we talked about these maximum residue levels, and I talked about the actual residue levels we found in our recent study funded by PCC. But from a steroid hormone standpoint, probably only like 1 to 10% of the actual amount that's in food and meat ever gets absorbed in the human GI tract. So that information also becomes very important. Other compounds like the beta adrenergic agonists, they're much more orally active, even in the meat samples. So maybe 80 to 90% of that can get absorbed. So knowledge for the average consumer and also the athletes, I think that's very important that they understand that many of these steroid hormones that are present in muscle cuts or meat are not easily absorbed in the human GI tract.
Well, that makes sense. I think this is really fascinating work, Brad. And we highlighted earlier that it's very transdisciplinary. And I think some of these collaborations can be really important when you're working on these transdisciplinary research projects. So can you talk about how you collaborate with other stakeholders, whether it's funding organizations like the PCC, other scientists, labs, people in industry, in the work that you do?
Yeah, so I attended my first PCC meeting in New York City in April of 2023, and I know there's another one coming up this coming spring. Hopefully share some of these results of this funded research. I've also been involved in the USADA Science Symposium. I was a speaker last fall at their meeting as well. So that's just great networking opportunities to get more collaborations with scientists that are interested in anti-doping.
I think that's phenomenal. And I know funding is important for researchers to be able to do the research they want to do and answer some of these big questions that remain. So, Brad, if we took away all of the barriers that normally hold you back and we gave you all of the resources and funding you could dream up, what is the one problem in anti-doping that you would most want to solve?
I've often thought about that, Marie. And what's interesting about how we use these compounds in meat production, one thing I've never really brought up is trenbolone acetate. We always combine that with estradiol 17-beta. So I've challenged some of the analytical people that are involved in anti-doping research. Is there a way that once we inject something like trenbolone in a cow or a beef animal, does it have a different, say, stable isotope pattern when it's bound to its receptor compared to just injecting some of these compounds directly into humans? I think there's so much we need to learn yet. It's obviously going to take a lot of resources, but I do think there's opportunities that at some point we can definitively say that, no, this trenbolone came from directly injected into a human compared to it was first injected into an animal like a beef animal, and then the meat was consumed. That is what I've been thinking a lot about lately, and obviously would take a lot of resources, but I think that would really advance our knowledge and help on the anti-doping front.
Well, I love this dream project, and I agree that this would definitely bring us closer to having some of these key answers that would help us answer some of these questions that come up in anti-doping related to this particular area of study. And we've talked about some different aspects of your work and some details in particular about your recent PCC-funded project. But if our listeners want to learn more, Brad, what is the best way for them to do so?
The best way is they can reach directly out to me via email. My email is Bradley, bradley.johnson@ttu.edu. But also I publish all of my research in peer-reviewed publications. And so they could do a Google Scholar, a PubMed search for some of these relevant articles as well. And also, like I had stated before, I was involved in the USADA Science Symposium. So I think if people were attendees there, some of my slides and other transcripts of that presentation would be available online at USADA as well.
Excellent. Well, Brad, we appreciate you sharing those resources. Listeners, definitely take the time to learn some more about the great research that Brad is conducting. And Brad, thank you so much for taking your time today to spend with us to tell us more about your work. It's been a pleasure to chat with you.
No, thank you, Marie. This was very enjoyable, and hopefully the listeners find this very valuable.
Absolutely. It's been wonderful to have you here. Listeners, wonderful to have you here with us as well. If you haven't already, it would be great if you could take a moment to rate our podcast and leave a review on your favorite podcast platform. Make sure you subscribe. And listeners, please note that the Anti-Doping Podcast features a wide variety of clean sport experts, and episodes may include statements and or viewpoints that do not necessarily reflect the views or values of the PCC. Thank you, and we look forward to seeing you on the next episode of the Anti-Doping Podcast.